Hello, welcome. Good day, everyone, and welcome to today's FIP digital event brought to you by the Industrial Pharmacy Section webinar series. The title of our session is Aspartyl, Asparaginyl, Beta-Hydroxylase, ASPH inhibitors for the treatment of cancer. FIP envision a world where everyone has access to safe, effective, quality, and affordable medicines and health technologies. Our mission is to support global health by advancing pharmaceutical practices, sciences, and education. So we are pleased to deliver this webinar as part of our ongoing effort to share relevant scientific and professional insight with the global pharmacy community. My name is Aini, and I am honored to be your moderator for today. I currently serve as the FIP, Early Career Pharmacy Group, or ECPG, Industrial Pharmacy Section Liaison. I'm joined by Dr. Matthew Cherian, Vice President of Operation and R&D at Revive Biotechnology USA, who will be co-moderating this session. We are truly excited for the discussion we are about to have and look forward to your engagement throughout the webinar. So before we begin, a few housekeeping notes. This session is being recorded and also live streamed via YouTube. After the session, the recording will be available at www.events.fip.org. Please feel free to submit your question at any time during presentation using Q&A box provided. We'll address them during the Q&A session. We'd love to hear your feedback. You can email us at webinars@fip.org. And if you are not yet a member of FIP, we warmly invite you to join us at www.fip.org/membership_registration. This is exciting news. We have new Zoom translation feature. So to access it, please first turn on caption in your Zoom toolbar, then enable the translation toggle, and then finally select your preferred language under my caption language. This allows you to follow along with live translated caption in over 30 languages. Here, what you can expect from today's session: first introduction, speaker presentation, Q&A, and closing remarks. So before we move into the presentation, let me briefly introduce today's topic and learning objective. ASPH, or aspartyl asparaginyl beta-hydroxylase, is an enzyme increasingly recognized for its role in cancer progression, particularly through pathway like Notch signaling. This enzyme is not typically active in normal adult tissue, but is often overexpressed in tumor cells, making it a highly promising therapeutical target. So today's session will help us to understand what is ASPH, how does ASPH modulate the Notch signaling pathway, and consequences of ASPH inhibitor activity. Now, it's my pleasure to introduce our distinguished speaker, Dr. Mark Olsen, PhD, Associate Professor of Pharmaceutical Sciences at Midwestern University, Glendale, Arizona, USA. So Dr. Mark Olsen is a senior medicinal chemist whose research focuses on designing and evaluating inhibitors of ion-dependent deoxygenases, including ASPH and FTO. He earned his PhD in chemistry from the University of Texas at Austin and completed postdoctoral research at MIT. Originally from Columbus, Ohio, Dr. Olsen has held academic appointment at West Texas A&M University and now at Midwestern University's College of Pharmacy, Glendale campus. So we are truly honored to have him with us today to share his expertise on targeting ASPH as a potential strategy for cancer treatment. So please welcome Dr. Mark Olsen. The floor is yours. Thank you so much.
Thank you, Heidi. The first thing I would like to say is it is quite a privilege to present my work to you. And I also want to thank Dr. Matthew Cherian for generously inviting me to give this presentation. So the title of my talk is Aspartyl Asparaginyl Beta-Hydroxylase Inhibitors for the Treatment of Cancer. And if you've never heard of ASPH before, don't worry. It's not commonly discussed. It's a relatively unusual enzyme for modulating the Notch signaling pathway. Very briefly, was already given a kind introduction by Aini. Basically, I grew up in the pharmaceutical industry where my father worked with Dr. Cherian back in Columbus, Ohio. And I'm primarily a synthetic medicinal chemist, although I also engineer antibodies and enzymes as well. First arrived at Midwestern University, I wanted to work on something different. And so I designed a family of molecules designed to target 2-oxoglutarate. And there are a number of iron-dependent dioxygenase enzymes which are involved in epigenetics. Some of them directly work on DNA or RNA, as in the case of FTO. Some are post-translational modifying enzymes for modifying proteins. And I had absolutely no idea what ASPH was when I first started this particular research program. I was primarily trying to target prolyl 4-hydroxylase. I designed a whole family of inhibitors. I let my fingers do the walking, and I submitted my molecules to the world expert in each one of these various enzymes. And to my very great surprise, one molecule and one molecule only had really good activity against ASPH, and that particular molecule did not have good activity against any of the other epigenetic enzymes that you see on the screen. And so I had to very quickly learn about ASPH and what role ASPH plays in the body. Oops. So briefly, ASPH is a transmembrane protein with a significant number of domains. There's a transmembrane domain. The catalytic domain is the extreme C-terminal domain. It turns out that there are a very, very large number of splicing variants of ASPH that do not include the C-terminal domain. And so there's a lot of erroneous information in the literature and in publicly available databases, for example, from the Broad Institute, on the expression of ASPH, because they look at the expression of the core protein and do not necessarily pay attention to the expression of the C-terminal domain, which contains the catalytic activity that we will be talking about today. So what exact reaction does that C-terminal domain do? Well, it hydroxylates very specific aspartic acid residues or asparagine residues in calcium-binding EGF-like domains. And it's not just any calcium-binding EGF domain. It needs to meet a specific canonical hydroxylation sequence that was originally identified in Denmark back in the 1980s. And it turns out that because of crystal structures performed by Dr. Schofield and coworkers in the UK, we have nice crystal structures, and there's an unusual conformation for the cofactor 2-oxoglutarate. And so we believe that this allows for the selective inhibition of ASPH. The hydroxylation occurs in a very specific domain and a very specific position in the calcium binding EGF domain. And so this is the core reaction that is performed by ASPH. What the heck happens? Well, basically, this particular enzyme has the ability to hydroxylate a lot of different calcium-binding EGF domains, and this domain is relatively common in the Notch signaling pathway. So you find the calcium binding EGF domains in all of the Notch receptors and all of the Notch ligands. And when you go in and you examine the exact domains for do they meet the canonical sequence for ASPH, sure enough, you find that the key binding domains for the Notch signaling protein are all classical ASPH substrates. And not only do you have the Notch receptors, but you have the Notch ligands that almost all also are potential substrates for ASPH, and most of them have been experimentally verified at this point. But you also have some proteins which are known to be Notch modulating proteins, such as the CRUM proteins, CRUM1 and CRUM2, that also appear to be ASPH substrates. So that's interesting, but what's the functional consequence of this? Well, in general, in the normal GI tissue, you have an interaction between Notch1 and Jagged1, for example. They bind to each other and you get the normal activation of the Notch signaling pathway. This is the canonical Notch pathway that people are familiar with. Fine. And that has to happen for the normal epithelial cells in the GI tract. Tumors inappropriately overexpress ASPH. It turns out that ASPH is highly expressed in the endoderm layer of the developing embryo and is very important in embryological development and for invasion of the uterine wall for development of the placenta. But in the adult humans, the expression is dramatically reduced. And tumors inappropriately overexpress ASPH. And so what we think happens is ASPH hydroxylates not only the receptor, Notch1, but it also hydroxylates the ligand, so you have a stronger interaction between the Notch1 and the Jagged1, so you get a more intense activation of the Notch signaling pathway. This is not good news, particularly for patients. The prognosis for cancer patients that have a strong ASPH expression in the tumor is notably worse in almost every cancer that we've looked at relative to tumors that do not overexpress ASPH. So it's involved in tumor proliferation, but even more importantly, and what I really want you to remember is it's linked to invasion and metastasis. So how the tumors are spreading, okay? So there is a little effect on proliferation, so I have to include that on the list because that's true, but the big effect is on the invasion and the metastasis. So if you're trying to target the Notch signaling pathway, one way to do that is, for example, is to inhibit the cleavage of Notch1. Another possibility is to develop an antibody which binds to Notch1 and prevents the Notch1 interaction with Jagged. This basically shuts down the Notch signaling pathway, but this causes a severe dose-limiting GI toxicity, as you'll see with gamma secretase inhibitors later in my talk. And so this is a serious problem. This entire issue, however, can be resolved by going in and targeting ASPH. When you inhibit the ASPH, you allow for the normal interaction between Notch and Jagged in the intestinal epithelial cells, but you suppress the ability to activate the Notch signaling pathway in the tumor cells. So this is a GI sparing strategy for targeting the Notch signaling pathway. I should also mention that these are not the only proteins which are hydroxylated by ASPH. These are the ones that are most interesting and most exciting and we have focused on first, but there are other proteins in the body involved in cancer signaling pathways, roboproteins, fibrillins that are also ASPH substrates as well. So we're focusing on the Notch pathway because we understand it the best and we focus the greatest amount of time and research energy on Notch, but there are many other effects by ASPH as illustrated on the next slide. So there are proteins which are involved in tumor angiogenesis. There are proteins that are involved in modulation of the Wnt signaling pathway. We've already talked about the Notch signaling pathway. There are proteins that are known confirmed experimental substrates for the TGF-beta pathway. There are proteins which are linked to activation of planar cell polarity, coagulation cascade proteins such as factor 10 are potential ASPH, known ASPH substrates. We have pro-metastatic factors, we have lipoprotein receptors, and we also have modulation of immune system proteins. I'm not prepared to talk about some of that work. We have some work in press showing the combination of ASPH inhibitors with immunomodulating agents and immuno-oncology strategies. Okay, so when this hydroxylation occurs, what happens? Well, there aren't very many crystal structures of proteins that have the ASPH expression because it's only found in mammals, and most proteins that are crystallized are produced in bacteria or in insect cells. However, when we do go and find a crystal structure that does have the appropriate hydroxylation, what you find is that you have an increased number of hydrogen bonds between the ligand and the receptor in the crystal structure. And so we believe that this is the origin of the enhanced interaction between the ligand and the receptor at the molecular level. So a quick summary for the ASPH biochemistry. ASPH is primarily expressed in embryological development, but not in adult tissue. Tumors inappropriately overexpress ASPH, resulting in enhanced migration and invasion. ASPH hydroxylation is very specific for certain calcium binding EGF domains and not all calcium binding EGF domains. The hydroxylation is not necessarily stoichiometric, meaning that ASPH hydroxylation is condition dependent. In some proteins, you may get 4 or 5 domains being hydroxylated. In other proteins, in other contexts, you may not get so much hydroxylation. We believe that the hydroxylation is involved in this protein-protein recognition, but these domains also bind calcium, and there is still the possibility that there is an interaction with the calcium binding function of the calcium binding EGF domains. All right, so I'm primarily a synthetic organic chemist. I have been interested in the scaffold of the tetranomids for a very long time, dating back to the 1980s. The specific inhibitor which hit ASPH, the very first molecule, was MO1-1100. It was our first molecule. It had a little bit lower potency, has relatively low toxicity. In medicinal chemistry, am a known fluorophile. When I have molecules and I want to enhance the potency, one of my very favorite tricks in my bag is to throw fluorines at the molecule. And sure enough, changing the chlorine to the trifluoromethyl group resulted in a dramatic improvement in potency. And we have demonstrated a significant amount of enhanced activity in a whole variety of animal models of cancer using the trifluoromethyl group. We have also gone and taken a look at the metabolic stability of the racemic compound and found that we can stabilize the chiral center by placing a deuterium there. And when we do so, we can separate the enantiomers. And when we separate the enantiomers, we get some very interesting effects. So with a racemic molecule, you have a half-life clearance with human liver microsomes of about 40 minutes. In mice, it's about, the half-life is about 26 minutes. When you place a deuterium on there, you notice that the half-life is increasing. When you resolve the enantiomers, one enantiomer is essentially not metabolized by human liver microsomes with a half-life greater than 60 minutes. But interestingly, you don't have such a strong effect in mice. This is a species-specific effect. And the beauty of this is that we can use the enantiomerically pure deuterated form of the compound potentially for humans, but for our animal studies, we can simply use the racemic compound with the hydrogen, which is much cheaper and much easier to work with. And so one isomer, which happens to be the active isomer, is the one that is not metabolized. The other enantiomer is very, very rapidly metabolized by the human, and is not of such interest in terms of human therapeutics. So stereochemistry matters. All right, so what do we actually see with ASPH? This is an example from a relatively early paper from 2014, where you see the overexpression of ASPH in hepatocellular carcinoma. In this particular data, which is from patients in the United States, the overexpression of ASPH was about 80%. Okay, when you repeat these same experiments, for example, in the People's Republic of China, where you have a much greater number of patients that are hepatitis B positive, that number jumps up to greater than 90-95%. Okay, in the United States, hepatocellular carcinoma has a more heterogeneous origin that is not so tightly linked with hepatitis B, but in East Asia, where there is significantly greater natural reservoir of hepatitis B positive patients, you find a much greater percentage being positive for ASPH. And so there's a direct link in terms of ASPH and hepatitis B positive cancers. In pancreatic cancers, we found that the percentage that overexpressed was 97%. As you increase the number of samples, one of the things that is really striking about the pancreatic cancer result is that not only do almost all of the tumor biopsies that we evaluate express ASPH, but they express very intensely. If you go and you take a look at NCI cancer cell lines from pancreatic cancer, you find that only about barely 50% of the pancreatic cancer cell lines overexpress ASPH. And one reason that we think that this may be true is in a plastic dish where you have infinite nutrients being provided by the experimentalist, there's no need for the cells to be able to invade and access oxygen or nutrients in the bloodstream. There isn't a requirement for that invasive capability, and so it gets lost in many of these pancreatic cell lines. And so you find significantly greater ASPH expression in tumor biopsies than you do in cell lines, and you find much greater impact in terms of how aggressive the cells are in vivo than you do even in vitro invasion assays, for example, Boylan chambers. So we have gone through a large number of cancers. Some of our early publications were on hepatocellular carcinoma. We have investigated pancreatic cancer extensively. There's work on lung cancer. I'm very excited about some new work that is in progress on pediatric osteosarcoma, where greater than 80% of pediatric osteosarcoma biopsies were positive and strongly positive for ASPH. We have done some work on breast cancer, ovarian cancer, as you'll see later in the talk. We've done a little bit of work in terms of prostate cancer, but not as much. And interestingly, we have some good preliminary results in certain canine cancers. All right, so what happens when you treat with an ASPH inhibitor? Well, basically what happens is you suppress signaling going through the Notch signaling pathway. So you have your control over here on the left. You have your downstream activators of the Notch signaling pathway, the HES1 and the HEY1. And when you treat with the ASPH inhibitor, you strongly suppress activation of the Notch signaling pathway. And this is statistically significant. And you get good results, in this particular case, in vitro. We have lots of data in terms of showing that cells that do not express ASPH have almost are almost not affected at all by the ASPH inhibitors. We very quickly moved ahead to some in vivo models because we know that the ASPH effect is significantly greater in vivo than it is in vitro. We used a particularly challenging tumor cell line called the focus cell line, which stands for Friendship of China and the United States. This is a cell line originally from the People's Republic of China that does have an inverted hepatitis B insertion in it. This is an entirely doxorubicin resistant cell line. So we treated with our ASPH inhibitor. And sure enough, we got results. Okay, so this is primarily an anti-proliferative type of model. So we see suppression of that. This is not incredibly impressive. Okay, when you have a tumor, for example, that has a very high expression of a kinase, for example, it grows dramatically more rapidly. And when you go in with the kinase inhibitor, you get a much more beautiful result experimentally. But sometimes those results do not always translate as well to the clinic. And so we're interested in the ASPH. And so, yes, we have an impact on proliferation, but as you'll see later in the presentation, we also have a really strong impact on metastasis and migration and invasion. So one control that we wanted to run was we took one of those pancreatic cancer cell lines that does not express ASPH at all, and we put in an empty vector in it, and we injected it in vivo. And we injected a first-generation ASPH inhibitor at a high dose, 50 milligrams per kilogram. And in this particular case, you don't see any effect on the parent cell line that does not express ASPH at all. It's not even statistically, you don't even have to use statistics to show that difference. Okay, when you take that exact same cell line, you overexpress ASPH, you see that it does grow more. Okay, this is an increase in the rate of growth of the tumor, but it doesn't blow your socks off. It's, again, not like cranking on a kinase. When you go in and you treat with the ASPH inhibitor, you knock it right back down to pretty close to where the cell line was without any ASPH at all. Okay, and so this is our very good in vivo demonstration of target engagement of these molecules in vivo. That wasn't good enough. We wanted to go in and take a look at the tumors from the experiment on the previous slide and go in and take a look and see what happened to the Notch signaling pathway when you treat with the ASPH inhibitor. And the data is a little bit data here, but you actually see suppression of your downstream signaling proteins of the Notch signaling pathway in HEY1 and HES1 for the tumors that were overexpressing ASPH but were treated with the ASPH inhibitor. So that's great. Cancer has been cured many times in rodents. Rather than doing a subcutaneous experiment, let's try and run something orthotopic to get a little bit better idea of, is this thing really going to work in an orthotopic situation? And the general rule of thumb with these ASPH inhibitors is that the more advanced and more difficult the model is, the better the result you get. And so, sure enough, this is an orthotopic experiment. This was, again, injected IP. And we got a significant suppression, not only of the tumor weight, okay, but also we got a suppression of the peritoneal tumor metastases. And this was what we were really looking for because we've been my collaborator, Dr. Jack Wands, had been after this anti-metastatic activity for several decades, and it was extremely gratifying to see the suppression of this metastasis, not just in an in vitro model, but in a real in vivo model and a relatively tough in vivo model. And again, this is with the focus cell line that's a doxorubicin-resistant hepatocellular carcinoma. All right, so going back to the subcutaneous model, we wanted to ask, all right, so what happens with the dose? Okay, and so we gave the compound that had the fluorines with enhanced potency, and sure enough, when you dose with DMSO or at 0.1 milligrams per kilogram, they're almost exactly the same. As you increase the dose, it turns out that there's no statistical difference between 0.5 milligrams per kilogram, 1 milligram per kilogram, and 10 milligrams per kilogram. Okay, clearly, the higher the dose, you have greater suppression, but statistically, they didn't actually separate from each other. Because the molecules are relatively simple and easy to synthesize, we typically just run at 10 milligrams per kilogram because there's no reason not to. When you look at those same results in a different way, what you see is that, yeah, you have an effect on the tumor weight going from 0.1 milligram per kilogram to 0.5 milligram per kilogram. You see a significant reduction, and when you boost the dose, you don't get that much greater effect in terms of the tumor weight. But you might also observe that, hey, wait a minute, Mark, you started out with 7 animals in the group with the DMSO. By the time you get to 0.5 milligrams per kilogram, you're at 5. By the time you get to 10 milligrams per kilogram, you're all the way down to 3 animals. What the heck happened? Well, the answer was we were getting cures in a dose-dependent manner. Okay, and so this was, a really tough cell line. This is doxorubicin resistant, and we're getting these results. We are suppressing the tumor weight. We're also, in some cases, actually getting cures of animals in these situations. Great. So what about other cancers outside of hepatocellular carcinoma? Here's data from pancreatic cancer. Pancreatic cancer strongly overexpresses ASPH. My collaborators were able to get results at a dose of about 50 nanomolar in terms of suppressing the ability of ASPH expressing pancreatic cancer cell lines to start to grow out of organoids. There was also suppression of invasion and metastasis in terms of those assays. That's great. But what happens in vivo? All right. And so here we're not dealing with IP injections. I had an army of pharmacy students. We packaged these into size M mouse capsules to try and make this as realistic as possible. We know our pharmacokinetics in mice. We have a very good half-life, mean retention time in mice. We dose them with these small mouse-sized oral capsules that you can buy from Torpak in India. And we were able to show that, yes, you did have a decrease in the tumor weight, but what we were really interested in was suppression of the metastasis. And sure enough, we were able to suppress the metastasis in a statistically significant way. It was a relatively easy assay to do because you simply do histochemistry on the lungs and you can go in and count the metastasis. And the beauty of this is that this is from a patient-derived xenograft model. Okay, so this was a patient which was taken from a human, was put into a mouse. It grew, proliferated in the mouse. The mice spontaneously developed pulmonary metastasis from this human tumor. We were able to then treat those mice with this drug packaged into a small size M mouse capsule at 10 milligrams per kilogram, and we were able to have a strong suppression of the metastasis. And so this was an absolutely great result. However, when you do a patient-derived xenograft model, you're inherently using an immunocompromised animal, and we know that ASPH has the ability to help tumors evade the immune system. And so we were really interested to see, is there a model that we could use using a syngeneic model, where we were using an animal with a normal immune system to see, can we get, in a slightly different context, can we get suppression of metastasis? And so we did that. And so we used a rat cholangiocarcinoma. Cholangiocarcinoma is a very, very aggressive cancer of the bile duct. We packaged the drug into rat-sized oral capsules. They were administered orally. Okay. at 10 milligrams per kilogram. And we had even greater suppression of the lung metastasis in this spontaneous model as well. And all of the downstream markers, sometimes it's very convenient to use MMP2 or MMP14 for invasion and migration. They are turned on by the Notch signaling pathway. Okay, and with treatment with this improved inhibitor, MO1 1182, we were able to get almost complete suppression of the lung metastasis in this immune-capable animal in the syngeneic model. And so this is a really great result, very encouraging for our strategy. A really good proof of principle. We've done this in other cancers as well. This happens to be an example of doing almost exactly the same experiment with breast cancer. There are a significant number of triple negative breast cancers that strongly overexpress ASPH, 50–70% of them. And we observed very similar results in terms of suppression of the metastatic capability of these breast cancer models in animals. All right, so that's great. We've got great results, but you can have really good anti-metastatic activity if you just make the animal really sick and you give it something toxic. What do we know about the toxicology of these compounds? And the short answer is, I wish I knew more. Other strategies for targeting the Notch signaling pathway, for example, an early gamma secretase inhibitor from Pfizer. Okay, you can see that the vehicle is on top, and as you give the drug, what you see is a decrease in the body weight of the animal, indicating that it's got toxicity, and you can measure that many different ways. And this is a well-known phenomena. It's caused problems with the gamma secretase inhibitors for decades at this point. When you treat with the gamma secretase inhibitor, you find this goblet cell metaplasia and hyperplasia in the GI tract, so you get the severe dose-limiting GI toxicity. And it's well known, and you get the same thing when you treat with certain antibodies as well. So what happens when you treat with an ASPH inhibitor is the very first question that I wondered about. We gave the drug at 50 milligrams per kilogram. We gave it in rats. We took a look at their food intake, and we saw no statistical difference. We looked at their body weight, and we saw no statistical difference. If anything, it was trending toward the animals had a little bit of an increase in body weight, but it wasn't statistically significant. We ran panels, all sorts of kinase panels. We ran 87 different anti-targets with the compound. We didn't find anything. Every drug has its dose-limiting toxicity, and that's going to be true for the ASPH inhibitors as well. But the dose-limiting toxicity is not the GI toxicity, and we haven't found it yet. We've looked at it a number of different ways, a number of different times. This particular data I can talk about. There's other data that I can't talk about. We keep looking for the dose-limiting toxicity, and it's been a challenge. You go in and you take a look at the small intestines, you do your immunohistochemistry, you stain for ASPH. You don't find any differences at all. Okay, and this is at 50 milligrams per kilogram IP given daily, every other day. So we still have a ways to go. That's really a core question for preclinical drug development. I still need to go out and chase down some money to try and enhance some of the preclinical toxicology models. But all the data so far is extremely encouraging. We've had very good activity against both subcutaneous and orthotopic models of hepatocellular carcinoma. We've had really good activity against a really tough patient-derived xenograft model for pancreatic cancer. We've had really good activity against a rat cholangiocarcinoma model, which is metastatic. We continue to explore the structure-activity relationship. We've demonstrated oral bioavailability. We've demonstrated suppression of metastasis using an oral dosage form. And we know that there are other proteins that we can chase down as well. We focused on Notch first because that's the very well understood and very well linked to this pathway, but there are clearly other pathways as well. All right. When you have a specific inhibitor, one of the very first questions that you need to answer is which patients are going to benefit. And so at MIT, I engineered antibodies. And so the very first thing I wanted to do after having discovered this family of ASPH inhibitors was to say, all right, I need an antibody so I can tell which patients are likely to benefit from these drugs and which ones aren't. The previous data was generated with polyclonal antibodies, mouse polyclonal antibodies. So I went in and I engineered a mouse, it's actually a rabbit monoclonal antibody. What we did is there is a crystal structure again from Christopher Schofield, and it turns out that the extreme C-terminal domain of the catalytic domain actually forms some direct hydrogen bonds with the substrate. And so we designed an antibody that not only allows us to do immunohistochemistry, but is also an inhibitor of the enzyme by binding to the extreme C-terminus that directly interacts with the substrate. Okay, sometimes people like to call this a neutralizing epitope. I just like to call it an enzyme inhibitor. So we designed the antibodies. It turns out that the regions that we are targeting are very consistent between both humans and canines. We got a reasonable affinity antibody. I would really kind of like for it to have been, single digit nanomolar. We wound up with, 144 nanomolar affinity antibody. Could be better, but it was good enough for these preliminary types of experiments. Okay, so when you have a brand new antibody, what do you want to do? You want to go and take a look at control. So here's a control where we actually stained, in this particular case, with an ovarian cancer tissue on top, and we did the isotype control on the bottom. And so sure enough, we get specific staining there. And then we went and did further investigations with ovarian cancer. And sure enough, our isotype controls are nice and clean, and we get nice strong staining in our tumor array in this case, this is stage 3 endometrioid adenocarcinoma. When you look at normal ovarian tissue, you don't see any staining at all. This is consistent with many, many experiments that we've done where trying to find ASPH expression is very elusive in normal body tissue. Okay, when you look at a benign tumor that is not very aggressive and does not invade or metastasize, okay, you see almost no ASPH expression at all. Okay, and so we have an antibody. This antibody is also a neutralizing antibody, so we can use this for certain mechanistic studies because we know exactly where it binds. And this is the antibody that we continue to use going forward, particularly taking a look at pediatric osteosarcoma samples. We're trying to spread out and take a look at a greater number of pediatric cancers as well, some Ewing's sarcoma, some neuroblastomas. All right, so very quickly in conclusion here, we have ASPH inhibitors. We've significantly enhanced the potency of the ASPH inhibitors. We've used these in a whole variety of different models, such as hepatocellular carcinoma, pancreatic cancer, cholangiocarcinoma, metastatic breast cancer. We have not identified our dose-limiting toxicity, but it's not for a lack of effort. We've demonstrated oral bioavailability. We've demonstrated suppression of metastasis in an oral dosage form. We focused on the Notch signaling pathway, but there are clearly other pathways to take a look at. For example, TGF-beta went. I'm very intrigued by the potential interaction with Gas6-Axel, but haven't yet found quite the right collaborator for that particular protein. And we have other compounds using fragment-based drug design that are alternate chemotypes that I have not spent any time talking about today. For acknowledgments, I've had an army of students here at Midwestern University that have worked on the synthesis of many of these ASPH inhibitors, and that work continues to this day. I've been very fortunate to have some great external collaborators, Dr. Jack Wands at Rhode Island Hospital, Dr. Anthony Lucci at MD Anderson, Dr. David Carpentieri at Phoenix Children's Hospital. I've had great support inside Midwestern University. I have had great interactions, particularly in Czech Republic, with Dr. Michal Šmihal and Dr. Ruth Tehezi from Biosev at Charles University, Prague, Czech Republic. And I would like to thank Midwestern University and Rhode Island Hospital for funding the vast majority of these studies. Some money has come from NIH. And last but not least, I would like to thank all of you for your kind attention.
Thank you so much, Dr. Mark Olson, for the insightful presentation. That was fascinating to look into the potential of the ASPH targeted approach. Now, we'd like to open the floor for question. If you have any question, you can chat through the Q&A box if you have question. Now, at this moment, we have some question. The first question, Doctor, could you please explain how specific ASPH in tumor progression, perhaps in percentage, compares with other established tumor microenvironment factors? Sorry, I cannot hear your voice.
Okay, sorry, I muted myself. My apologies. So yes, ASPH is a surface expressed protein. It is also expressed in the endoplasmic reticulum. It has the ability to hydroxylate a variety of different factors in the stromal environment. The specific question that was asked was about the percentage.
Mm-hmm.
In general, most of the work that we have focused on has been taking a look at what happens with prognosis. So when you look at ASPH expression, you can see that the prognosis drops very significantly with increased expression. With those with decreased ASPH expression or no ASPH expression, the prognosis is dramatically better. What percentage? Depends on exactly what tumor you're talking about. In the case of hepatocellular carcinoma, we see the expression in general, I'll just ballpark it at 90%, okay? We do not find any ASPH expression in many of the leukemias or lymphomas, okay? There may be a few unusual, spectacular exceptions, but in general, we find relatively high percentages in solid tumors and very little in terms of hem-onc, for example.
Thank you for the answer, Dr. Mark Olsen. Now, next to the question. Aspartyl, asparagine beta-hydroxylase belongs to a large family of enzymes with similar domains. Has its specificity been validated? Available studies? Are there any documented side effects associated with its inhibition?
I will take the second question first. In terms of documenting the side effects, we have tried really hard. We have tanked the animals up on very, very high doses of the ASPH inhibitors, including some of the advanced generation ASPH inhibitors, and we have completely failed to find side effects. Now, that doesn't mean that there aren't side effects to be found yet. There's always something else to be investigated, but we don't have a good answer for you in terms of exactly what are the side effects at this point in time. The first question, the first part of that question, could you repeat that for me?
Yeah. Has its specificity been validated? Available studies?
Yes. So one of my collaborators is Dr. Chuan He from the University of Chicago, who has developed a whole series of assays for this iron-dependent dioxygenase family. And so we have taken these compounds and we have incubated them with all of the near family members, and we have found absolutely no inhibition at all with FTO, with TET1, with the other related enzymes. We have gone and a number of people have asked us very carefully, are you sure that this is not a kinase inhibitor? Because when you get good results, many times people start to wonder about an off-target kinase inhibition activity. And we've looked, and we've looked, and we have completely missed. We have not been able to find any kinase with any reasonable inhibition.
Okay, thank you. Now we will talk about resistant mechanism. As with many targeted approaches, cancer cells often find ways to escape treatment. Do we have any insight into potential resistant mechanism that might develop against ASPH inhibitor, and what strategies are being considered to anticipate or counteract these challenges?
That is an excellent question.
Sorry, Dr. Mark Olson, your voice we cannot hear.
Uh,
Yeah,
okay,
good.
can you hear me now?
Yes, good.
Okay, so that's an excellent question. I cannot necessarily tell you how the cancer cells will become resistant to ASPH expression. We have not run those experiments, but what we have done is we have taken a look at cancer cells that are resistant to PD-1 and PD-1L checkpoint inhibitor antibodies. Okay, these are commonly used, and we find very high ASPH expression in many of the resistant checkpoint inhibitor tumors that are available. We have been able to show RNA and protein expression in them. And so we know that ASPH plays a role in resistance for checkpoint inhibitors, but in terms of what is a role of resistance to an ASPH inhibitor, I can't tell you. I don't know yet.
Okay.
I'm sure that for every effective therapeutic, there is a resistance mechanism. Okay, so I'm sure that it's out there. Exactly what is it in this particular case, I can't say.
Okay. Thank you so much for the answer. And then about monitoring treatment response. In terms of tracking whether a therapy is working, are there any promising tools such as imaging technique or circulating biomarkers that can help monitor a patient response to ASPH inhibition in real time?
The short answer is you can find ASPH RNA in exosomes in certain cancers. So there is at least a theoretical possibility of being able to examine progression by taking a look at exosomes. We have looked at combining, putting a PET tracer on the ASPH inhibitor to get a theranostic so that we can look at where is the drug going and how is it interacting with the tumor. But in terms of being able to monitor the progression at this point, I'm on the medicinal chemistry side. I'm primarily involved in the design and the synthesis of the new drugs. I don't necessarily have partners in terms of how to go about monitoring that. We have gone through lots and lots and lots of animals, but in terms of monitoring it, normally we just sacrifice the animal and count the pulmonary metastasis for the most part. So you can't really do that with a human. So no, I haven't gotten that far. It's a good question.
Thank you so much, Doctor. Now, there is question about manufacturing challenges because some ASPH targeted therapies like monoclonal antibodies or vaccine fall under the category of biologics. These are powerful tools, but can be complex to produce. On your opinion, what are some of the practical challenges, whether related to manufacturing, storage, or delivery, that need to be addressed to ensure clinical success?
In general, monoclonal antibodies are pretty good in terms of de-risking them for manufacturing production. Your concern with the biologics sometimes is cost. Sometimes the concern is in terms of allergenic side effects, some things like this. But in general, monoclonal antibodies are very well behaved and we can scale them up. We've got a bunch of different systems for doing that. And so that is clearly scalable. The small molecules, I have an even better answer for you because the synthesis of even the deuterated compounds is a 3-step synthesis. Okay, and so we're able to get to these molecules very quickly, very easily. And if you don't need the deuterated compound, if you can just use a racemic compound, it's only a 2-step synthesis. And so that's the good news. The bad news is that one of those reactions involves cyanide. Okay, and so there will be certain challenges in terms of scaling that up. But, in the context of toxic molecules these days, um, cyanide is relatively well known, the risks we know, and it's not really that bad. And so being able to have access to a 2-step synthesis, and for an enantiomerically pure compound, you add one more step because you've got to deuterate, okay, basically using base. And so the scale up is very attractive and very realistic.
Thank you, doctor. Now we have last question. Can ASPH inhibitors be used in combination with immunotherapy?
Yes, and so we have a paper in progress with my colleagues in the Czech Republic, and it is coming out, and I can't quite talk about it yet. And the answer is yes. And we believe that we should be able to suppress a significant amount of the tumors that recur after treatment with checkpoint inhibitors if you co-administer with an ASPH inhibitor. Okay, and so we've got the preliminary results there, and I really, really want to get a good set of experiments working with some of the cutting edge checkpoint inhibitors that are currently approved by the FDA. But in terms of your general immunotherapies, yes, and it can synergize with them as well.
Okay, thank you so much, Dr. Mark Olsen. I think that is for me, and I will handle it back to Matthew if you have further question and also for closing.
Well, Mark, thank you very, very much for a very informative, path-breaking technology and presentation. Thank you very much for your time. Very, very interesting. We look forward to hearing from you in the future about future developments that you are doing in this area.
Thank you very much, Dr. Cherian. It's been my privilege to give this presentation, and thank you very much for inviting me.
Thank you so much.